Our guest today is physician scientist Stephen Quay. Stephen Quay is the founder of Seattle-based Atossa Therapeutics, Inc., a clinical stage biopharmaceutical company developing novel therapeutics and delivery methods for breast cancer and other breast conditions. He has founded 6 startups, invented 7 FDA-approved pharmaceuticals, and holds 87 U.S. patents. Over 80 million people have benefited from the medicines he invented. When the pandemic first began, what signs indicated to you that this may have been a lab incident?
So I think it was to look at the location of where viruses like this live, because a virus will typically be in a bat colony and it'll go 40 or 50 kilometers from its location. But Wuhan is about 1,000 kilometers from the cousins of SARS-CoV-2. And so that fact, and then coupled with the fact that some of the most intense research on coronaviruses in the world over the last 2 decades, over 60% of all the research in the world is done in Wuhan itself.
Now, given the information that we have now, what are the odds, do you think, are that COVID is the result of a lab incident?
So I've looked at this from a sort of a mathematical way and looked at all of the evidence and then used a process that's commonly used called the Bayesian analysis. And you get to over a 98% probability that this was not from nature and that it came from a laboratory.
What is your response to recent claims that raccoon dogs started the pandemic?
Yeah, so the evidence that people are talking about is the fact that there's a market where many early cases occurred. We knew early on that there were wildlife in this market that are susceptible to SARS-CoV-2. In fact, SARS-CoV-2 can infect a wide range of mammals. So the finding of the animals there was not necessarily probative. And then when the genetic material that the animals in the specimens were looked at did not have the virus within them. So I think it was a very interesting hypothesis, but the hypothesis was disproven by the absence of SARS-CoV-2 genetic material in the specimens that had the raccoon dogs.
And as far as raccoon dogs goes, are there any signs that this animal has carried the virus or transmitted the virus or in China?
That's a very interesting question, and in fact the laboratory experiments with raccoon dogs shows it's not a very good host. It's actually difficult to infect and transmit from one raccoon dog to the other. And so it doesn't meet the criteria of an intermediate host, even in a theoretical sense.
Could you elaborate on the new evidence that there were 2 lab incidents?
The evidence there is within the genome. If you look at a process that takes you back to what would be the first genetic virus, and you do the timing on that, it appears to be in the September-October timeframe. There also is a burst of cases in December. We're very aware of that, a lot of discussion around that. So those 2 events, the genetics that take you into the September-October timeframe and the human hospital incidents in December could support 2 separate introductions into the community.
And what kind of scenarios would involve that? What would cause that sort of situation to occur?
It would be 2 independent laboratory accidents. There's actually precedence for this in SARS-1, where a single laboratory had 2 separate accidental releases into the community. SARS-1 is very interesting. It began in a wildlife market, and it ended with laboratory-acquired infections, lab leaks as they sometimes are called. So the same epidemic had the bookends of the current theory for SARS-CoV-2.
So it had both theories in reality. Interesting. Now, is there evidence that some speculate that it started with a failed vaccine program?
Okay. I think that the evidence I have rules that out. There are some very specific changes you need to make in coronaviruses if you're going to use them as vaccines. It's a bit complicated and in the weeds, but the simple answer is they tend to recombine a great deal. There are some genetic ways to block recombination that have been recommended or actually been patented for making vaccines with coronaviruses. SARS-CoV-2 has none of those elements. So if someone were making a vaccine, they would know the proper way to make a coronavirus vaccine, it doesn't have those elements. So that's my evidence that it did not start as a vaccine.
Is there any evidence that this was a bioweapon of some sort?
So that's an important question, but also a very dangerous is the wrong word. But I think you need to be very careful when you look at that. So unfortunately, like maybe perhaps many kinds of research, civilian-based research, military-based research can often be the same process in the laboratory, and it's only what's in the minds of the people doing it or on paper of what their goal was to know the difference. For example, asymptomatic transmission is extremely rare in new respiratory viruses. It's one of the features of SARS-CoV-2. It's one of the things that fooled doctors for almost 2 months. So if there was evidence of research of that, could have been done to understand how asymptomatic transmission could occur from a wild virus, or would that be a purposeful feature of something that had a more nefarious intent?
Now, what are some developments coming down the road that might show us more evidence of the origin of COVID?
So I think we have to assume that we probably will never get what you might call a forensic examination of the laboratories or even the market by Western scientists, Western investigators. So we're in a situation where, as I sometimes describe it, we have a spouse who's disappeared. We have a spouse who looks highly suspicious, and the district attorney has to say, okay, I don't have a body, but I have the circumstantial evidence that I think I can put this spouse away for the murder of their wife or whatever. And that evidence that's presented in a trial. We do go to the most extreme, criminal cases with only circumstantial evidence, and we can successfully move that through the judicial process. I think that's where we're going to have to end up in terms of this process.
And what steps could be taken to prevent this sort of thing from happening again, if it was a lab incident?
So, and I think that's probably the more important thing we need to go to, which is if this virus came from a laboratory and it's spread around the world, one way I look at it is, 2 natural viruses, SARS-1 and MERS, affected about 10,000 people. Killed 1,000 in one case, about 3,000 in the other. So 10,000 people is the capacity for a natural spillover. We now have a virus that has probably infected 2 to 3 billion people. That may be a measure of the difference between a laboratory gain-of-function virus and a natural virus. So having said that, stopping or putting guardrails around this kind of research, I think is the most critical thing we can do now.
So there's evidence that the Wuhan Institute of Virology is conducting new gain-of-function experiments?
Yes, and this is probably the most important motivation for me to try to put guardrails around this kind of research. The work that I and my colleagues have done over the last 3 years has allowed us to basically do a forensic examination of the kind of research going on inside any laboratory in the world over the last 2 years. During that process, we found 3 separate incidences of research, gain-of-function research, synthetic biology research involving a MERS virus, which is approximately 30% lethal, involving an influenza virus, which is approximately 49% lethal, and the Nipah virus, which is between 80 and 90% lethal. So this is synthetic biology with all of the tools that are used to move genes around and to juice up viruses. All of this research going on inside the Wuhan Institute of Virology identified since January 2020 when this pandemic started. So we have just barely survived a 1% lethal virus, SARS-CoV-2. These are civilization-ending viruses based on my analysis of the Black Plague, which was about a 25% lethal event. It's a 500-year setback for civilization if one of these viruses gets out of a lab. This is really important, and I think, I'm frustrated because I try to look at policy things, but so we have all of this energy around climate change, which is an important issue, but is a really slow process. It's like a 100-year process here. And this is something that could happen in a week, a month, a year, a decade, but it will happen if we don't do anything about it. Because accidents, they're all accidents, in one way or another, I think, but it will happen. And so then we've got a virus spreading that's 30% lethal. And I'm actually doing a detailed study on why this would end civilization. Because we have food, energy, police, and fire, and hospitals. And if you break the supply chains of any of those in a singular fashion, or some combination of the four of those, we're done. And we're back to 1850.
Thank you for coming today and speaking with us at Taiwan News.
Thank you. It's good to be here.
Thank you.